This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 136, recorded on June 3rd, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV, the podcast all about viruses. Joining me today from Western Massachusetts is Alan Dove.
Good to be here.
Beautiful day, isn't it?
Yeah, and I'm happy to report that the tornado that came through here missed us.
That's right, you had it out there in the west as well. I heard it was east.
Yeah, it landed in Springfield, just north of us, and cruised through Munson and did a whole bunch of damage and knocked power out to thousands of people. And we just got some thunderstorms, but
it's unusual to have tornadoes in Massachusetts, isn't it?
It is. Technically, they can happen anywhere in the country, but it's a little odd here.
Not in Kansas anymore, Alan.
No.
Also joining us today from North Central Florida, Rich Condit.
Hey, except I'm not in North Central Florida.
That's right. Where are you?
I'm in Newton Highlands, just outside of Boston.
Oh,
okay.
In the basement of my daughter's house.
Well, if another tornado comes, that's a good place to be.
Exactly. Yeah, we were here when that stuff came through. We were downtown that evening, and it was a genuine Florida rainstorm. It rained.
Cool.
But fortunately, we were distant from the tornadoes themselves.
Yeah, you guys are both in the same state today. How about that?
That's right.
How about that?
Same state, same tornadoes. All right. No 85 degrees and blue skies, Rich.
No, but we got blue skies at 67. That's almost better
bad
at this point.
Also joining us today for a second time on TWiV is my colleague here at Columbia, the Departments of Biochemistry and Microbiology, someone I've known for over 30 years, Steve Goff.
And I'm happy to be here. Hi, Vince.
Welcome back.
Yes, all the way from behind the wall from next door.
Steve is right next door to me every day. Well, actually, you travel a lot, so you're not here every day. But today we have Steve Goff because we are talking about XMRV. A bunch of papers came out this week.
No.
And actually, there was a paper of Ela Singh's a few weeks ago, which we didn't talk about, so maybe we'll get to that one as well. And to give us credibility, we have Steve here.
Okay.
Because Steve is one of the finest virologists I know. He's a very level-headed guy. He doesn't go out on a limb, and he's usually right. So that's
wow.
Yeah. Not that you aren't, Rich.
Oh, no, well.
But he's in the retrovirus field.
Yeah.
Now, don't set him up or anything here.
Actually, I'm usually wrong, okay? But
No,
that works well for some people.
No, Steve he's conservative. You'll say, hey, this means this. And he goes, well, I'm not so sure about that. And he'll tell you why. And he's right. So it's good for the XMRV discussion. So the last time you were with us, Steve, it was the beginning of XMRV, and we talked about it. Now it's a lot later, and a couple of pretty substantial papers have come out. This week in Science, there are two papers we're going to talk about. One from the labs of Vinay Patak and John Coffin on the recombinant origin of XMRV, and then a second paper from Jay Levy's lab, no evidence of XMRV in previously positive patients, and then a very interesting movement by the journal Science to get the original XMRV paper retracted. So we'll talk about all that.
Right. So since that first time that we were here, it's we do know a little more, at least.
Mm-hmm.
Maybe we don't know everything, but we know a little more than we did.
You came from a retrovirus meeting just recently at Cold Spring Harbor, right, Steve?
Right. So these papers were discussed and presented, and there was actually almost a whole session on XMRV. So we could talk about those as well.
Okay. What is your sense of work on XMRV in general? Is it, since the original 2009 paper in Science by Lombardi et al., has it increased, decreased, stayed the same?
Well, so first of all, that's not the first paper. That's
right.
a first paper linking it in principle to chronic fatigue syndrome.
Right.
But I would say, since then, the numbers of papers and the numbers of people talking about it at least have both gone up. So it's quite a lot of activity. Now probably plateauing, peaking, and I suppose dropping in the future because most of the questions seem to have been resolved.
That's right. So the first discovery of this virus was 2006 by DeRisi and Ganem, right?
Right, and that was in the context of prostate cancer.
Right.
So the first early years of XMRV interests were really centering around its potential role in prostate cancer. But then things really got turned up a lot with the potential link of the paper that you mentioned from
2009,
Lombardi on chronic fatigue syndrome. Right.
All right, so let's talk first about the Patak Coffin paper. You know both of these investigators pretty well.
Very well.
In fact, John Coffin you've known for years, right?
Right.
He actually looks a little like you.
Right.
No, you just both have beards, that's all.
I've been introduced as John Coffin several times. I always feel flattered when that happens.
He's really good, isn't he?
Yes, he is.
He's another really smart retrovirologist. The retrovirology field seems to attract really smart people.
Or gray bearded people, I don't know.
David Baltimore was another one, who we both worked for. Anyway, the first paper has to do with this cell line called 22RV1, which is a human prostate tumor cell line, right?
Right.
It's been known for, I don't know, a couple of years now that it actually makes XMRV, and that is almost identical to all the XMRV sequences that we have from people, right?
Right. And in fact, when that was realized, that the sequences of the virus in that cell line are really virtually identical to the reported sequences of the things that were being isolated, that was a worry. I mean, that raised flags in a lot of our minds because that should not be, in that retroviruses evolve rapidly and are subject to mutation rapidly. So as you made independent isolates of a similar virus, you would have thought they would vary. The sequences would vary.
There's just 4 different nucleotide differences between this virus and the human isolates.
Right, virtually none.
Out of 8,000 bases, right?
Right.
Right. Whereas if you isolate HIV from a single patient over time, you see more change than that.
Right. Or even within a person. You would see more variation than that. And if you isolated Maloney, the mouse leukemia virus that we study, from different mice, you would see more variation than that.
So I guess the question was, this cell line, it produces XMRV. Is that because the original prostate tumor used to make it was infected, or was there something else? I guess that's what they're looking at here.
So that was the subject of this paper, this Coffin and Patak paper. And they did a really amazing detective work.
Tell us what they did. You're best qualified to do that.
So they basically were able to track down the original samples of the original tumor, or close to the original as they could get, which came from,
1993, I think.
93, yeah. And then various isolates of it. So this is a typical line in that it was ultimately prepared by explanting a tumor from a patient and then transferring it in mice. In
1992.
92.
In nude mice.
Now, why is that done?
And that's done because a lot of human lines simply aren't easy to culture. So you put them in, you put them in culture, and they just don't survive. They don't replicate well. They need growth factors that we don't still know, or they need conditions for their growth that we still don't know. But a mouse provides all that. So a mouse is a perfect way, a nude mouse in particular that doesn't have an immune system to reject it, is a perfect host to grow a lot of human tumors. So that's done in the 70s, 80s, 90s, still today.
Ultimately, this winds up at some point in time explanted from the mice and grows in culture?
Right. So what you find is that after many passages through mouse-to-mouse transfers, eventually the cultures, the tumor lines adapt. They probably suffer more mutations, but they become able to grow independently, which is a moment of rejoicing for the lab, usually, because now you can handle the line much more easily in culture. You can clone the cells. You can study them in
your mouse budget goes way down.
Exactly. So that happened with this line, this R22RV line, over the course of time. And Coffin and Patak were able to go back and recover intermediates in that process, and then test them all for the presence of various virus sequences. And what they found was that the very earliest isolates they could get their hands on were virus-free. That is, they at least did not have any replicating retrovirus in them. And later passages did. So something had happened in the course of this passage in mice, and then in culture, that in fact that resulted in their infection. And John Coffin is an expert of the endogenous retroviruses in mice in general. And so he had been looking for years for the source of what we now think of as XMRV. There were various very characteristic features of the virus that he could use to ask, does this provirus in mouse have it? Does this provirus in mouse have it? And he looked at dozens, maybe hundreds of mice. And no mouse that he looked at, neither strains in labs or wild mice, had any single provirus that was a perfect match to the thing we now have that we call XMRV. So that was a puzzle, and he more or less over the years said he would keep looking. And what he found finally, though, was that there were nude mice that had proviruses that had half of XMRV. And Vinny Patak, looking in a similar way, found proviruses that had the other half of XMRV. And so they called these endogenous proviruses in mice pre-XMRV1 and pre-XMRV2. And they are proposing in this paper that what has happened in the nude mice in which the tumor was being propagated, that a recombination event occurred. That's not crazy. That's not rare. Retroviruses recombine very frequently. And so rather than delivering intact a single copy of a virus from mouse to human cells in the mouse, they proposed that recombinants were transferred from the mouse to the human tumor line.
Right. They point out that you'd need 6 template switching events to get this to happen, and you get an average of 4 per replication cycle.
Right. So even 6 is not crazy. Although
right.
Pushing it, I mean, there's really just one major recombination event in the middle that is required, and then there's some sort of cleanup work at the very 3' end that seems to have gone on.
Yeah. In fact, these papers are all open access, so you can. There's a very good figure where they diagram the 2 pre-XMRV 1 and 2,
right,
and how they crossed over to form XMRV, or theoretically. So if you do this crossover on a computer and make XMRV from pre-1 and 2, you get a virus that is a few bases different from all the clinical isolates.
Virtually identical to this new virus that's being studied.
So let me clarify this. You don't find XMRV in any mice, either lab or wild.
Right.
But you find pre-XMRV in lab mice.
Right. So the likely source then was this strain of nude mice. Right. Interestingly, in the detective work, there's no records of exactly what nude mouse was used in the early propagation of this tumor. People don't keep perfect records. Stunning, isn't it? But because they had the tumors, and because the tumors had mouse DNA in them, once you've grown a human tumor in a mouse and explanted it, it's got mouse cells in it. They were able to deduce the likely nude mice that were used in the process, and so they were able to see that yes, these nude mice had these 2 halves, if you will, or these 2 proviruses that could donate half of their genomes to make XMRV.
Looking at this figure, it looks to me as if there's only one of these guys that has both of the previruses, the HSD mouse.
It's rare that a mouse has both. That's correct. There's a fair number of mice that have one, and a fair number of other mice that have the other. There's only very, very few mice that have both. And I think
I'm basically wondering whether we're talking about a recombination event that, whether the 2 previruses were transferred independently and you get a recombination event in the cells, or whether this is happening in the mice and infecting the tumor, or what.
Right. So I don't think we know that for sure. The history, the prediction would be as follows. These XMRVs, these xenotropic MLVs, which is what they really are in mice, don't replicate in mice as a rule. Right. Their envelopes are such that they don't work in mouse cells. They work in other mammalian species. That's actually the basis for the term xeno, xeno meaning other. So they're from a mouse. They were once in a mouse, but the virus that is in the mouse genome doesn't replicate in mice. So they're kind of frozen, intact in the genome. But when it's really an amazing thing. When you expose the mouse to a human cell by putting a human tumor line into it, those cells are exquisitely sensitive. So the extremely low amounts of virus that the mice have derived without viral replication from these endogenous viruses then replicates in the human cells. So that's what happens more or less very frequently when you put a human tumor into a mouse. So in this case, we would say the recombination event would most likely happen at that time, at the time that these mouse viruses are, for the first time in the mouse's history, successfully replicating in these human tumor lines. Mm-hmm. So you would need, in principle, these two defective viruses, defective from the point of view of the mouse, to be getting into the human tumor cell line, presumably a cell, a human cell, in that tumor at the same time, and then they would recombine.
But that could be done either serially, at different passage numbers, one at one passage, one at another passage, or simultaneously if the tumor was being harbored in a mouse that had both previruses.
That's right. You're right. So you could, in principle, pick up one, still leaving it there in the human tumor, and then later pick up the other one, and then they would recombine. That's fair. Good point.
And you would essentially select for a virus that could survive in the human cells.
Right.
And in particular, grow in prostate.
Yeah, and that's a very interesting point that Coffin and I were chatting about the other day. That one of the things we've known about, and I'm very curious about still in my lab, is the fact that XMRV, as we know it, has an incredibly potent tropism for prostate cancer cell lines. Its promoter is really hot in prostate lines. And we think we've tracked that a little bit to being a function of the androgen response elements in the 5' region of the virus that drives transcription. So what you would suppose here is that this acquisition process selected, just as Alan says, for a virus that grows really, really well in human prostate lines.
Do you know whether it grows well in, I don't know if you can do this experiment, normal prostate tissue?
We haven't really done that. We've done the best we could, which is sort of just survey a handful of other human prostate lines. We've only looked at less than a half dozen. And it tends to grow really well in most of them. Mm-hmm. And it grows really, really poorly in other human lines that we've looked at, like HeLa or 293. It's 100-fold different, at least. As a matter of fact, the xenos, as a rule, the xeno MLVs, tend to be really wimpy, feeble viruses. Their promoters don't work very well. And I think maybe that's because we just don't know what cell lines we should be growing them in. We would have all liked to have worked on them in the 70s ever since, if we could, but they're so difficult to get to grow that people said, I'm going to drop it and work on
So you can take an XMR-free prostate tumor line and infect it with XMRV, right?
Indeed, and it grows really well.
But it doesn't do anything to the cells that you can detect. Is that correct?
That's right. Like most retroviruses, they're pretty benign for the cells. Cells don't die. There's no cytopathic effect. And your only sign of being infected is if you look with things like reverse transcriptase assays or with antibodies.
So this could travel silently from one culture to another in a lab.
Easily. And that's true of the mouse viruses in mice cells. So a lab that Vincent and I grew up in, the Baltimore lab, for example, had most of its work done with 3T3 cells, a very standard mouse line that, if treated well, is free of virus. But it's very easy to get them infected, and it's invisible to the naked eye.
Well, and we talked about a paper, I think a couple of months ago, where some researchers came across a recombinant virus that had been artificially generated in another lab years earlier under biosafety level 2 conditions and should never have gotten into their lab because they'd never worked with that cell line or that virus or those people. And it just showed up in their cell cultures.
Yep. Yeah, and that's not very surprising. So it's easy to get these retroviruses around. If anybody in the lab or on the floor even is working with one of these viruses, there's a fair chance it's going to end up getting into a lot of different cell lines, if that cell line is a good host for the virus, and if nobody looks. Because,
so when you exchange cell lines with somebody, you're exchanging cell lines with everyone they've ever exchanged cell lines with.
It's a little bit like sex, isn't it, in that regard? Yeah.
Yeah, you never know what you're getting.
Reminds me of the phage days, people who worked on T1 basically weren't allowed in your lab because it would just come in and trash all of your cultures.
There's a great story that somebody wanted to get a T1 phage strain from a guy and he wouldn't send it. So they took the letter. In those days, people actually sent mails. They took the letter that said, "No, I won't send it to you." And they soaked it in water and got the phage they wanted off the letter because they figured it was on his fingerprints.
So Coffin and Patak say that XMRV arose from a recombination event between two endogenous MLVs that occurred between 93 and 96 in a nude mouse with this CWR22 xenograft.
At Case Western Reserve University.
That's right, CWR. And all of the XMRV isolates reported to date are descended from this one event, contamination. And it's hard to escape that because the likelihood that this same recombination would have happened somewhere else, maybe in the 80s or 70s, is pretty remote. I think they said 10 to the minus 12.
Right, vanishingly small.
You are a millionfold more likely to win the lottery on a given ticket.
So I think all the circulating isolates of a XMRV or its anybody like it, if they've been sequenced and they are exactly like the virus that we were talking about, that's the indication that it's really a contaminant.
So this xenograft was done at the Cleveland Clinic, and I understand that this CWR22 cell line, wait a minute, is that the name of it?
We call it 22RV1.
Sorry.
Yeah,
For many years, it wasn't even known to make virus.
Right.
So you can imagine that the virus would contaminate lots of specimens, right?
Yeah, it would be easy for that to happen.
So when DeRisi and Ganem got prostate tumors from the Cleveland Clinic in 2006, which they used to discover XMRV, they were somehow already contaminated with either virus or proviral DNA, right?
Yeah, I mean, I guess we don't know the details, but the line was widespread and widely used in many labs that were prostate cancer labs. It's an interesting line. There aren't too many human prostate lines to study. This one, in the course of its derivation, now we know became infected with this relative, normally invisible virus.
It's pretty conclusive evidence, in my view.
Amazing,
right?
Just amazing.
This paper, of course, we all saw this paper coming because it was presented at a conference and the video was online and all that. But for me, it's never really real until I see the data. And not just on a slide projected on the screen for a moment. So for me, this was really just mind-blowing to see this answer come out, which is totally not what I was expecting, at least. And it's just fascinating.
The figure that Vincent pointed out that lines up the two pre-viruses.
Yeah.
Yes.
And anybody could go across there and draw the recombination events.
Column 2, 3, 4, 5, 6, right?
Yeah.
And
I was not aware that. I mean, I knew that retroviruses recombined and that they were relatively recombinogenic, but I wasn't aware that even on average you can get as many as 4 crossovers. That's pretty heavy traffic.
Yeah, so it's during reverse transcription that this happens, and of course it's an aspect of the fact that retroviruses have 2 copies of the viral RNA per particle.
Right. Okay.
So and that's also a consequence of the fact that the enzyme doing this, the reverse transcriptase, as it's making DNA from RNA, has this very, very low processivity. So it pops off one template and reinitiates on the other one. And I think the numbers are that this happens on average in the realm of 1, 2, 3, 4, 5 or so times per transit.
And processivity means its ability to go forward is low, right?
Or it's no. Well, I mean, in this context,
It's attention span.
It's processivity on a single template before falling off and reinitiating on the other template.
So I might get myself in trouble here, but if I recall the normal reverse transcription process, there is an obligatory template switching event after priming, is there not?
Well, there are two obligatory jumps, as they're called, from one to the other, indeed, that are ordered or directed at occurring at a very specific point. And so we can talk about those in great detail. So you make a small molecule called -strand strong stop, and you stop making it. It's a stop because you run out of RNA to copy at the cap. And so then that molecule has to jump. So that's just a part of its life to have to do that. And there's another jump that happens on the other strand in a somewhat similar way.
Right.
But,
so this idea of template switching isn't a completely crazy idea.
Right. It's not bad from the point of view it's even needed for the virus. But these jumps in the midstream, as it were, are done with homology from one point on one genome to the homologous site on the other genome. It's probably good for the virus to do this too, actually. Because, for one reason, if you hit a break or a tough spot, the way around it is for the virus to switch to the other template. So retroviral RNAs, retrovirus particles, are actually quite resistant to damage from that point of view. You can you can successfully get a virus that will work with both of its RNA genomes heavily broken, because it will hit a stop and go to the other one and get over the break.
So the idea here is that pre-XMRV 1 and 2, we had two RNAs in a single particle somehow.
Exactly.
And then when that was reverse transcribed, it just randomly jumps, and the one that will replicate in human cells is the one that propagates.
Exactly.
Right? Because
exactly.
the virus, it doesn't know where to jump between XMRV 1 and 2 because there are no breaks as far as we
It's
know.
happening randomly.
Yeah. Right, but this happens often enough that you eventually hit on a solution, and this is how viruses mate.
Right.
Exactly. So that you eventually have a progeny virus that is capable of surviving in this new environment, and of course that's the one that's going to grow out.
Right. And this happens. This is a big deal in lots of settings. I mean, so in HIV, there are areas of Africa that have clades, strains of HIV-1. You can have clade A in another area, and we'll have clade C. And where these epidemics overlap geographically, you can see recombinants appearing, and so you'll get HIV clades A and C hybrids, and they often will have altered biological properties. Whatever grows best will grow out.
Right.
In fact, pre-XMRV1 is in fact a broken virus, right?
Right. It's defective, apparently.
It has some mutations that would prevent making an infectious virus, yeah.
So, and most of the endogenous copies that mice and we humans carry are defective, in fact. In fact, humans, as far as we know, don't have a single functional copy of any of its old endogenous viruses. There's a wonderful story there about, if you've already talked about, the HERV-Ks.
Yeah, we talked about how Paul Benash resuscitated one of them.
Exactly.
Right. So here there are other related xenotropic MLVs in mice, but this one is the one that matches XMRV.
Or these two
halves.
yes,
right? These two halves.
Right, exactly. There's about a dozen or so, as I recall, XMLVs in most mice.
Okay. So at any other time, a different one could have arisen by xenografting, right? But it's just this particular one, and it somehow contaminated everything.
Right.
And you'd predict that as these types of passaging experiments are done, this could happen again.
Oh, yeah, absolutely. Sure. And it has happened. That is, in the very early days of passaging human tumors in mice, as retroviruses were hot early topics, people looked and they said, wow, this human tumor has apparently a human retrovirus in it. And people were very excited in the 70s. And then they looked harder and said, oops, this was not a human virus. This was a mouse virus that particular human tumor picked up from that particular mouse. So it's not at all the first time that this has happened.
All right. Let's move to the other paper that was published along with it. This is from Jay Levy's group, and this is more like all the other XMRV papers that are looking for the virus in a variety of ways in patient samples. So here they have two groups of patients. The first one they call P1, 41 CFS patients from Sierra Internal Medicine, Incline Village, Nevada. And those of you who recognize that name know that Dan Peterson works there, and he was on the original 2009 science paper and provided many of those samples for that paper. So he has here 41, or in this paper, 41 patients. 37 had been previously tested for XMRV by either the WPI, the Whitmore Peterson Institute, and Peterson in that institute, by the way, is Dan Peterson, who provided the samples for this paper, or a commercial lab called VIPDX in Reno, Nevada.
Which is the commercial arm of the WPI.
That's right. 26 of those were reported as being XMRV positive and 11 negative. Then these blood samples were archived diagnostic specimens, so they could just pull them out of the freezer. And the second population came from the same practice, and the subjects were selected basically on having a XMRV positive test.
From the same lab.
Same lab. 29 patients, 26 of whom had been positive for XMRV, and at least one of 3 assays, PCR, virus isolation, or antibody. And they basically ran through all these samples by PCR culture, looking for antibodies in the blood by ELISA, and they were all negative.
So the difference between these two populations is that the first one is archived serum samples. The second one is fresh samples.
They got fresh samples,
blood.
Yeah.
And in fact, there's an overlap between those two pools. There are some of the patients for whom they had both archived samples and fresh samples. I forget the number.
Yeah, in fact, it says here the chart review indicates that 53% of the P2 samples, so a chart review of the 41 patients that's in the P2 group revealed that 19 had 2 blood samples taken on the same day by the phlebotomist.
Same phlebotomist.
And one was submitted to VIPDX and the other to the Wisconsin Viral Research Group, which is on this paper. And in the original paper, the science paper, some of those were positive, and here they were all negative. So
all this is important because a subtext here is that if there's contamination, where did it happen?
Right.
Is it the phlebotomist? Is it what's going on?
Right. So in this paper, those positive samples where they did the double draw were all negative in this new paper.
Right.
So the implication of the paper is that the patients themselves are really negative,
right?
And that when you get fresh samples, or even if you look at the clean old samples, they're negative.
And they did careful controls for contamination too.
Right, which is not trivial, by the way.
Right.
That's really important.
Yeah, if we get to Eli's paper, we'll talk about that more, I think.
Right.
An interesting thing what they did here, they addressed this issue of very little genetic variation between all these samples, and they said they passed VP62, which is the virus derived from the Ganem derisi.
Right, the original prostate cancer isolation.
And they passed it in cell culture, and they get lots of mutations, more than they've seen in all the patients.
Right, so that just says this is a virus, it's a real virus, and when it replicates, it mutates at the normal level that we are all used to expecting. It's not some weird virus that can't mutate.
In fact, Greg Tower had published that a while ago. He said in his lab he saw more variation on the virus coming out of 22RV1 than in all the patient samples.
Right. So this is the norm for these viruses, and
which is too bad, really, because if somebody had found a virus with this perfect fidelity reverse transcriptase, you could make a lot of money off that.
Right.
The other thing they did is they took sera from CFS patients and mixed it with virus, and it inactivated it to certain extents, healthy subjects and CFS patients. And they say this shows that it would be really hard from an MLV to establish infection in humans because there's some kind of an activating factor in serum.
Right, maybe complement. They're not. Yeah. I guess they don't know.
Yeah, they say,
it's in the serum of CFS patients and healthy subjects.
Yeah,
right.
They say when you pass the virus through human cells, it becomes less sensitive, which they say because then it's already seen human complement, and so that would be expected. So they say that this virus is not infecting people, and it's not causing CFS, and maybe there's some other infectious agent, but it's not this one.
Right.
In fact, their last sentence is, since an activated immune system has been observed in CFS patients, the possibility of another infection agent being associated with this illness merits continual attention, which I think is reasonable.
Right. And
True. Although
true.
it's worth noting that an activated immune system with similar changes in cytokine markers occurs in just about every chronic condition that anybody's checked in.
Right.
So it doesn't show that there's an infectious agent. It just this chronic inflammatory state that you see in CFS, you see it in patients with depression, you see it in patients with obesity, you see it in whatever. It's immunology's check engine light. It comes on all the time, and it provides no useful information.
Could be autoimmunity too, right?
Sure, could be an environmental toxin.
Yes, many possibilities. But maybe all of this has heightened awareness now, and people can look at this more.
Right.
In more detail.
I think over the years, the chronic fatigue syndrome has always raised the potential that there's some virus in there, and many viruses have been brought up over the years as possible candidates for being the causative agent. Sure. There's been talk about herpes viruses. Boy, I can't even remember the list. A long list.
Yeah.
Right.
And this is the latest, but I don't think any of them have held up.
I think maybe one possibility is that any infection can trigger an aberrant immune response in these patients for whatever reason. That's what we have to find out. All right, so the other thing I wanted to discuss, and I think we should talk about Ila's paper next, but first, on the basis of these two papers in Science, it's my understanding that the editor of Science asked the WPI group to withdraw their paper.
Right.
Right, because I think there's now many papers saying that the chronic fatigue syndrome link is not being supported by anyone.
Right.
So retracting that Lombardi paper seems reasonable to me.
And they said, of course, no, we're not retracting it. So then the Bruce Alberts wrote what's called an editorial expression of concern. It's a paragraph or so saying why we think this paper has problems, and they attach it to the paper as published. So when you go to see the paper online, you will see this editorial expression of concern.
Right.
Did you ever encounter that before, Alan?
Yes. Yeah, this comes up occasionally. And this whole thing has stimulated a whole conversation about how frequent are retractions. And I guess it was last week, Rich's pick of the week was Retraction Watch.
Yep.
Yeah. So there's a blog that posts this type of information and kind of tries to learn from it. The editorial expression of concern it's sort of the editors really think this ought to be retracted, but the authors are not doing it. So we're going to put a disclaimer on the thing, and it's one step shy of the editors withdrawing the paper themselves.
Mm-hmm.
So the editors, of course, do have the ability to do that, and may at some point in the near future with this. But at this point, they're saying, there are serious problems with this earlier study. We don't want anybody to, not that anybody could have missed the news on this in the field, but in the off chance that somebody just starts with a literature search on XMRV and pops this up and then decides to extend the work, they want to put a red flag on it saying, look, if you want to extend this work, you better start by checking every single thing in it. You can't assume that you can just work from here.
Right.
The last paragraph of this editorial comment is interesting. The U.S. National Institutes of Health is sponsoring an additional carefully designed studies to ascertain whether the association between XMRV and CFS can be confirmed. We'll maybe talk about that. We've talked about it before. Science magazine eagerly awaits the outcome of these further studies and will take appropriate action when their results are known. So that's a sort of a...
That's Bruce Albert saying we'll pull it when we need to.
Right.
So my feeling is that a paper should be retracted if it's fraud. If it's just wrong, it's going to be corrected. As we've said over and over, science is self-correcting. Because here now, all the other, the Harvey Alter study should be retracted. The PC, the Prostate Cancer Associations, because they're all the same virus. Well, no, the low Alter is not, but the prostate cancers are the same virus, and it's a contaminant clearly. So there
have been a lot of retractions that were not due to fraud, that were due to error.
Really? So that happens also? Yeah.
It does happen. It has to be such a serious error that it completely undercuts your confidence in the whole data set from the paper.
Okay.
Or undercuts some key aspect of it, so that, as I said, they want to make sure that if somebody looks this up in the future, they're not going to assume that the data in here are valid and then try to extend them.
Yeah. Okay.
That what you would need to do is go back to square one. First, try to get these same results yourself, because we don't know that these are valid. We suspect they're not. And then you could extend from there. And so retractions have been made for that. In fact, the Wakefield paper that we've talked about, they did turn out to be fraud. But before we knew that, the other authors on that paper had voluntarily retracted it.
Okay, so we should mention that this study that they mentioned, the NIH study, is being coordinated by Ian Lipkin.
Right.
Yep.
Do you know what that involves, Steve?
I'm not totally familiar with all of it, but I've talked to Ian Lipkin about it. So as I understand it, Tony Fauci basically asked him to try to validate the testing in various labs that were purporting to be able to see XMRV. And the scheme was that a number determined by Ian that is about 100 to give power to the test. 100 samples were going to be sent to Ian blinded. He was going to codify them, mix them up, and send them back to the labs of origin and ask them to repeat using their own assays for XMRV, and then let him know. And so he was going to basically say, did they get it right?
So the labs involved were WPI?
I don't remember. It was several labs.
Some others. CDC, I believe.
There's one, the CDC, the WPI, and it may be Harvey Alter's lab. It's somebody at the NIH, I think, or the FDA.
Maybe Rossetti. I'm not sure.
Maybe.
Yeah.
By the way, I forgot to mention the Levy paper. They also found extensive contamination of various reagents with mouse sequences. Let me pull up that paper again. And he says he thinks the low alter results could be explained by that sort of contamination.
And to be fair, that's the latest in a chain of reports that show that. That would include work from Greg Towers and the Ela Singh paper as well. So when you look hard, that's what you find. You find there's mouse DNA and everything.
I had a question about this. Now, some of these studies that find positives are not actually amplifying the whole virus. Is that right?
That's right.
Amplifying bits. So the contamination that we're talking about doesn't necessarily have to be virus around. It can be mouse DNA with provirus in it. Is that correct?
That's exactly right. Yep.
Right. People are using primers that would amplify, that would give you a positive result if it's XMRV or if it's
pre-XMRV1
some of these related viruses.
Yeah, that's what Coffin said in the paper, that he thinks a lot of the assays are actually picking up pre-XMRVs.
Right, or just general DNA. Other XMLVs that are not even that similar to XMRV. Because
not all the PCR signals get sequenced.
Right, exactly.
Right, and there are I think it was Towers who pointed out that there were some disturbing similarities between low and alters viruses and the ones that you can isolate from commercial PCR reagents.
Right.
Yeah, here's the passage I wanted to find. Levy and et al. Detected MLV sequences not only in 3 out of 5 Taq polymerases that use monoclonal antibodies, that's for PCR, but also in 9 of 17 other monoclonal antibody-containing reagents used in research labs, including antibodies to CD4, CD8, and CD14. Sequencing of these PCR products revealed a high degree of homology with known MLV sequences. From lab strains, they most closely resembled the MLV sequences reported by Low and Alter.
Right.
Right. I mean, I think any monoclonal antibody, which after all comes from a mouse, would be very hard to prevent from having mouse DNA in it.
To quote Eric Delwart again, Vincent, DNA is really a problem. It's everywhere.
That's right.
Yeah.
How providential that was when we talked to him last year.
Really.
We had no idea.
I'll never forget that quote. That was great.
Let's just briefly go over Ela Singh's paper because we haven't done it. This was published at the beginning of May.
Right.
And she took blood from 105 patients, 200 healthy volunteers. These were bled by a third party, and 14 of these patients had been positive in the original Lombardi et al. Study.
Right. She had tracked down the same patients that were in that study.
And she told us on TWiV it was really hard to get this information from Judy Mikovits. Okay, so they did PCR
and that's not the only hard thing in this study. This was a really impressive paper.
Yeah, she really was very thorough. She did 4 different kinds of PCR, looking for antibodies in serum by ELISAs, looking for virus in plasma, all negative.
Doing this cell culture assay. Yeah.
Right.
Culturing for 6 weeks, nothing.
And in between cell culturing assays, she sterilized the incubator with UV light and Clorox, I think, and just really excruciatingly thorough work.
And they also found mouse sequences in Taq polymerases.
Yes.
To try and understand the results of Harvey Alter again. So now I felt this was a really strong study, which clearly showed there was no XMRV in these samples, but it was roundly criticized by many as not duplicating exactly the studies of Lombardi et al. So, I mean, I think you can never exactly duplicate, right? Study, because first of all, it's in a different lab, and that in itself can make a huge difference, right? So that was, in my view, an important paper, but many thought it wasn't powerful. However, in retrospect, it's a really carefully done study, and it showed that these patients were negative. And now we have the fact that this is a recombinant virus, which really overshadows it, I guess, in many ways. Right?
And also this thing about it's not a true replication is a red herring. Because if the virus is in there, you should be able to detect it by a variety of techniques. And if you can only detect it using the super extra magical primer set and buffer conditions and moon phase and whatever else that was done in the previous study, then folks, it isn't there.
Right.
I mean, it's something else you're detecting that needs those special conditions, because the virus, if it's there, you should be able to detect it with any of a variety of PCR primers. You should be able to find the antibody with any of a variety of antigen probes. And these studies that have been done, these follow-up studies, especially this one.
Yeah, no, I think the Singh paper, in anticipation of the demand that you really recreate as much as you can, the conditions of the first studies really went over backwards to do that. And I think they really did a good job. So I think she's really got the right assays, primers, everything, and it wasn't there. So
yeah, as I was reading this paper, I kept being reminded. I've been to a couple of Passover Seders, and there's this thing Dayenu. You know, it would have been enough. And as I was reading through, it, okay, Ela, Dayenu. You know, it's just you could stop there.
Before we have a bunch of XMRV emails, which I'd like to get to, but before we do that, Steve, what happened at the Cold Spring Harbor retrovirus meeting in the XMRV session, besides these 2 papers, which I guess were presented? Anything else of note?
I think there were basically confirmations. There were many of the players were there, Greg Towers, Ela, and others. But basically there was a series of 3 or 4 abstracts essentially saying, yeah, we looked in this cohort and that cohort, and it wasn't there. There was the talk of the recombinant origin from the Patak and Coffin labs. And then there were some, less controversial aspects of the nature of the XMLV reverse transcriptase, of the XMLV protease.
Because it is a real virus.
Yeah, it's a real virus. You know, we think of it as another particular example of the family of xenotropic MLVs, and it has some interesting properties in culture. But I think the important high-profile part was the failure to find it in humans.
What do you think next year's Cold Spring Harbor retrovirus meeting will bring in the way of XMRV? Will it be diminished, you think?
Yeah, I think the crisis is over at some level. There might be a handful of papers on any of its novel features, whether it's the transcription promoter or there's some interesting things about the protease. but maybe there's some work on the receptor, which is an interesting molecule.
But those people at Case Western went to the trouble of creating it. You might as well work on it.
Yeah, and I think most of what you found out about it would be relevant and true for the general XMLVs.
The prostate cancer thing still has to be cleaned up.
Yeah, that's an interesting aspect.
And I think that
right.
my sense is that the original authors, or some of the original authors in those studies, are on their way, are working hard to get to the bottom of that, find out what happened.
The very original isolate from prostate cancer stories. Yeah. And I guess that, if we're closing up on this event, there'll be some, I suppose, further probes as to whether there's any virus like it in any human prostate samples. Are there some real underlying the contamination issue that we're sure is here in most cases? Is there anything real in patients underlying that? Are there some rare xenotropic MLVs lurking? And so there's some need to still look for that and know if those are real. And today, fortunately, we have the tools to figure that out, because if you now have some putative isolate from a human of something you think is an interesting retrovirus, the sequence will tell. If you sequence your new isolate and it's virtually identical to what we know as XMRV clone VP32, rather, well, forget it. It's a contaminant. But if you get something novel, well, then that would be pretty interesting.
A number of people online have said that the Lipkin study shouldn't go forward. It's spending money, and we know XMRV is a lab contaminant. What do we think about that?
I disagree. First of all, the money's already budgeted.
Well, they could give it back to Steve Goff.
No, I think this is a substantial study, and Tony Fauci asked Ian Lipkin to do it, and he darn well better finish the job. And there was some controversy about allocating that quantity of money for what would essentially be a follow-on study at a time when a lot of people aren't getting their grants renewed. So now that the money, I don't know how much of it has already been spent, but if Lipkin is saying that he's expecting to publish this by the end of the year, you got to have a few months for review and all that. I'm guessing that a good portion of the work is already done and the money's already spent. So I don't think there's really any point in discussing should you pull the plug on it.
Well, and if your description of that study is correct, an important component of that is that the WPI itself is doing some of these assays.
Right. And
yes, and the other thing that it could do is, I'm not certain of this, but it may give the legal authority for the FDA to tell them to stop.
Mm-hmm.
That maybe that's true. That might have been a reason for Tony Fauci to fund this.
It's a true large-scale, comparison study. Right now, the way the WPI/VIP diagnostics test is set up, it's operating under a stipulation or a loophole, depending on your perspective, in the CLIA regulations, for something called a laboratory-developed test, which says basically if you're a clinical lab and a licensed clinical lab can develop its own tests and market them, and all they really have to do is have a protocol and follow it and follow some basic practices. They don't actually have to do a rigorous testing initially to determine their false positive and false negative rate and all that sort of thing. So the Lipkin study is providing that rigorous testing. And if you have a lab-developed test that is subjected to that sort of analysis and it fails, then I'm pretty sure you have to pull it off the market.
Mm-hmm. So maybe that is something we'll be seeing in the coming year.
Yeah.
Well, I can imagine, too, that given how politically sensitive this whole thing is, that if all of a sudden decided to stop this study midstream, you'd get all sorts of cries of government conspiracy to squash the information and all that. I think you got to finish it.
Yeah.
Well, we have Ian coming next week on TWiV. He's going to talk about something else, but maybe we could slip a question in.
I doubt he'll answer it.
Yeah.
We can try,
I certainly wouldn't if I were in his position.
Well, he could describe how the study is being run, at least.
That's true.
Right?
Yeah, how it's being designed.
Because right from his mouth, but...
Right.
All right, let's do a few emails. These are all on XMRV. The first is from Valerie. What are the implications of the new gag sequences WPI has been publishing for the debate about XMRV? And she provides a bunch of NCBI links. Explicable as lab contaminants, explicable as lab recombinants. I'd really, really appreciate your insight about how these sequences might affect the debate.
They are explicable as VP62.
Have you looked at these, Steve?
I have not, but I gather from what I'm hearing, they're too close to 62 to be a real variant.
In fact, Vinay Patak sent me a tree and said they're all VP62.
Yeah.
22
Yeah,
RV.
when these came out, I went and did blast searches on them, and they're first of all, they're just gag sequences. They're not whole viruses, so you're only looking at a piece, but it's one or two bases different from VP62 or one of the other sequences. They're all clones.
The expectation was that these would be very different and could say then this is a virus that's been circulating in people, but it's not the case, right?
Right.
All right, the next one is from Joshua. I think that anyone interested in XMRV should be familiar with previous episodes of Scientific Controversy, and a great introduction is Nobel Prize winner Irving Langmuir's talk entitled Pathological Science. And he sends a link. The lessons he learned in 1954 are still completely valid today and far less emotional to learn from. I also think anyone who wants to report on XMRV should read Pulitzer Prize winner Gary Taub's wonderful book, Bad Science, which covers the cold fusion fiasco of 1989. As above, virologists can learn from the mistakes and successes of physicists from 20 years ago. And he provides a link for that as well. Love your podcast, Joshua. Thanks, Joshua. Those are two good links.
Yeah.
Check them out. Next one is from Lance, who read my virology blog post on the recombinant XMRV story that was just discussed, and I thought we'd read his questions. Two questions. One, how did this recombination event arise? As I understand it, these two viruses exist only as integrated provirus, and pre-XMRV1 is replication deficient. Does that mean the new virus, XMRV, was generated by the action of the pre-XMRV2 Pol gene on pre-XMRV1 genome, for example? I don't know enough about XMRV replication, but a discussion of this on TWiV would be great.
Yeah, well, that's almost what we said. I think that's the right scenario. A tidbit to know about these endogenous viruses is that even if they're defective, the mouse can express these integrated DNAs to make particles. If the gag protein is still functional. That's a requirement. But defective genomes can then be packaged into those particles, and recombination can happen during that reverse transcription process that we said.
Okay. All right, the second question is, what implications does this have for xenotransplantation? Could you speculate on the potential for the introduction of a new retrovirus through a xenotransplant to cause a replication-deficient HERV, for example, to become replication-competent, or for a novel virus to be generated?
Yeah, that's very likely, very easy to imagine. Most of the HERVs are in the human endogenous retroviruses that are sufficiently distant from the mouse ones that they're not likely to easily recombine as two mouse viruses would. But that's an issue that we've talked about, thought about in papers and in the lab for a long time. I think it is likely that a defective mouse virus replicating in human cells could easily recombine and pick up pieces of HERVs. You can even imagine rescue of defective genes from the HERVs. The repertoire of HERVs that are in our genomes include many pieces that are functional.
So we use pig tissues for putting into people, right? Pig heart valves and that sort of thing.
Right, and pigs have PERVs. That is, they have pig endogenous retroviruses, as they're called. And so the people that are thinking about organ transplants have worried about this. Yeah. It's a real concern.
So in the pigs, are they replication competent?
I don't think most pigs' PERVs are replication competent. There may be some that are.
But they don't have to be, right?
They don't have to be, exactly.
Yeah, they could be.
Right. I know there's been some work that has shown that at least the PERVs that people have looked at are easily inactivated by the human serum, kind of the way XMRV is by human serum. But that doesn't necessarily allay fears of giving somebody a virus by putting a pig heart in them.
Right. No, there's people, or Jonathan Story has looked at the PERVs. There are families. There's PERVs A, B, and C, and they even know that these use different receptors. So there's really quite a body of literature now about that.
And you'd have to be worried if you're writing your grants on that some congressman would get a hold of your proposal on PERVs.
Right.
Yep. Oh, yeah.
Totally misunderstand it.
Many thanks again for all the great work in the growing collection of excellent podcasts. What will be next? Who knows? Thanks, Lance. Tate writes, I have a question about XMRV contamination, with this argument getting a lot of traction in the CFS patient forums. The question involves exactly what constitutes a distinct virus. Some patients, and apparently the WPI, are claiming that the sequences found by alter low are simply variants of XMRV instead of being distinct from XMRV. Lots of discussion involves mentioning the sequence similarity of XMRV to alter low's PMLV being around 96% or so, whereas variants of other viruses that are considered to be the same virus can be 80% or less. I think I might have it figured out vaguely, and it has to do with how the sequences cluster on a phylogenetic tree. But if you would be willing to discuss this, it would probably help a lot of people understand the issue a little clearer.
Right. Well, I think there are no hard and fast rules. There's no simple single number that you can put on this to answer that question. So you really do have to look at trees and try to think about what the likelihood of a virus being derived from another one is. You know, if you had to put numbers on it, you'd say something that's 99% or 99.9% similar might well be a contaminant. Things that are 95% or 90% or 80% are more likely to have been more distant from each other and have diverged through many rounds of replication, not likely to be an immediate contaminant, at least.
So can you explain how the phylogenetic tree works? I mean, what are you doing there to say that these viruses are related?
Yeah, so if you have 20 viruses that you're putting on a tree, there are programs that will do this in the most parsimonious way, but you essentially arrange them in order of proximity, similarity to each other as best you can, and then the branches of the tree that you use to connect them have links that reflect the number of divergent bases between them. So guys that are very far apart will have branches that are very long. Guys that are very close to each other will have branches that are very short on the tree, and it's not rigorously that this branch reflects the evolutionary history exactly, because those trees that are designed based on different genes have somewhat different branchings sometimes. But it's a fair approximation of what could have happened in the evolution of a family of viruses.
So you don't want to just look at the percent difference. You have to construct this kind of phylogenetic analysis.
Yeah, it helps a lot, and it gives you a feel for what the range is in the family that you're talking about.
Which I think Greg Towers actually did in one of the retrovirology papers last year, right?
Mm-hmm.
Yeah, and one of the talks at Cold Spring Harbor had such a tree, and when you look at all the isolates of the XMRVs, they're all virtually flat. That is, the branches between them are minuscule, and you look at the branchings from that to any other mouse virus, the branches are huge. Right. So they really, cluster in a tiny spot in the space.
Okay. All right, Tate, hope that helps. Next one is from Marcia. She has a series of questions. 1, if the results are due to tests picking up widespread contamination, why aren't the results always 100% positive?
Well, if we were all infinitely careful, she'd be right. But, the levels of contamination that we're talking about here are down to the realm of molecules, literally. And it's really hard to get stuff down that perfect.
And contaminants by their nature vary.
Yeah, I think in the Singh study, she showed that she had some contamination of water, but it was so low that it wasn't reproducibly detected.
Right. She would do 20 samples, and 5 would be positive one day. If you did the same 20 samples the next day, a different 5 might be positive.
And that was the one she ultimately traced to the pipetting robot, right?
Might have been, yeah.
There was one. Wow. There was one. I remember the paper mentioned that they traced the problem to a piece of laboratory equipment that had been used for XMRV related stuff months earlier, and apparently some quantity of the virus still persisted on it.
Yeah, this was true of at least in one case for a microtome that was used to slice tissue samples. And a few months before, somebody had sliced a mouse, and then 6 months later, you're slicing human samples. That'll do it.
I mean, the problem is that the numbers in the 2009 Science paper, 67% of CFS patients, 3% of normal controls, they make sense if this is a virus causing the disease. But if it's contamination, why would it be so black and white? And that, I don't think we can explain it unless we go in and identify the source of the contamination in that laboratory.
Right, and it's important to note that the samples did come from two different places, at least. And well, now we know from the Levy paper that if you retest the original samples, they come up negative. But it's entirely, even if there was an attempt to blind them, it seems unlikely that they could have been completely blinded samples, because one set was from the blood bank and the other set was from a clinic.
Yeah.
We've talked about the source of contamination potentially being human lines that are propagated in various labs, and I think that may be right, or viruses that come from those cells. But I'll tell you, a real likely source is just clone DNA.
Yes.
Because as soon as you've made milligram large quantity preparations of a plasmid in your lab, there are nanograms of it everywhere. Right. It's really hard to get rid of it.
This all reminds me of John Coffin's rap about a drop of blood in a swimming pool.
Yeah.
Drop of mouse blood in a swimming pool, you basically contaminate the whole pool.
Yeah.
Yeah, you take out 50 microliters and you get a PCR positive for mouse DNA. So her next question is, if contamination is so widespread, how do you account for study results where no XMRV was found?
They were careful.
Yeah, they're very, very careful.
Really careful.
Yeah, and if you read Ila's paper, you've got an example of a lab that was contaminated and then took extreme measures to trace and find the contaminant and eliminate it, and now their results all come out negative.
Right.
If WPI says they did not use the contaminated line in a new lab, which is 22RV1, and have been testing weekly to guard against contamination, what else could they done to prevent the alleged problem?
Well, you have to take real extra measures everywhere.
Read Ila's paper.
Yeah, Ila's got a playbook for that.
Regarding the subsequent study that didn't show positive for the explicit variety of XMRV referred to in the October 2009 study, but did turn up more than one closely related virus, that would be low alter. Shouldn't subsequent studies be looking for those related viruses in the same way HIV testing looks for multiple HIV variants?
I think they are.
Yeah.
People have used the low alter primers, right?
Yeah, I think those primers are capable of amplifying either.
If the virus is difficult to find in blood but easier to find in tissue biopsies, why aren't researchers looking more at tissue biopsies? I don't think it would be difficult to find volunteers.
I do.
I don't know that it is easier to find in biopsies. Do you know that for certain?
I don't know.
Depends on what microtome you use.
Maybe.
Yeah, exactly. That's a source of contamination, yeah.
Yeah.
Maybe this is too obvious an idea, but given all the controversy, shouldn't there be at least one follow-on study that tries to duplicate WPI's methods exactly to see what happens? I got through on the phone once to Dr. Mikovits. Surely it wouldn't be that difficult for a researcher to call up and ask to meet with her or some of the other researchers there to understand their methods better.
See the Singh paper and the Levy paper.
Yeah. I mean, I think that's, you do want to try to do that, and I think the Singh paper has done really a pretty heroic job of doing it.
But that's the Lipkin study, as I understand. The WPI will be able to use their own methods to examine the blinded samples he sends them.
Right.
And by the way, Ila on TWiV said it was very difficult to get samples from Dr. Mikovits, if you go back and listen to that episode. So it's not so easy. And finally, what do you make of the handful of ME/CFS patients whose condition has improved on antiretrovirals?
I'm guessing this is just not enough cases to really.
Well, it's just not a controlled trial, right?
Yeah, and there is a low but documented spontaneous remission rate. So somebody could start taking antiretrovirals, then they start getting better, and they would have started getting better anyway.
Right.
And there's also a huge problem in every single clinical trial, regardless of disease, called placebo effect.
Yep.
Right.
And if you're taking a drug that has dramatic side effects based on a theory that you sincerely believe in, this placebo effect is huge in a case like that.
I think you can't conclude anything from these anecdotal reports because it is not a properly controlled clinical trial.
And if you look at the discussions among people who are boosting this as a solution, nobody who has a negative response is going to feel comfortable going online and saying, "Hey, it didn't work for me."
Right.
So there's a ratcheting effect in the whole discussion.
I mean, we know of anecdotal cases where that's been the case also, that people have gone on antiretrovirals, they didn't get better. And
what was the quote we ran across a while ago? Multiple anecdotes don't make data.
Right.
Yeah. All right, the last one is from Nicola. Dear TWiV guys, as perhaps you know, Judy Mikovits claimed for some time that she had sequence diversity showing that the viruses isolated from CFS patients were consistent with an infectious virus. Now that she has published a number of sequences in GenBank, she appears to have dropped this claim, as it does not feature in her reply to Science. I don't know what this reply is. I haven't found it. Now she is saying in this reply that she has shown that patients have antibody diversity, which proves they are mounting an immune response to a diverse group of viruses with MLV ancestry. And she quotes from this letter, the reactivity demonstrated to spleen focus-forming virus in the original 2009 paper was competed by the rat monoclonal antibody, which detects all known xeno, polytropic, and achotropic MLVs. This again suggests that we have in fact detected more than one strain of human gamma retroviruses in these patient samples.
No, it doesn't.
Yeah, I don't think that does.
Right.
No, it says that you can compete off the antigen with this broad-spectrum antibody, which is
not enough.
Not probative at all.
Yeah. He says, I have two questions. Does this constitute evidence of a diverse group of viruses? So no. Could these be cross-reactive antibodies or antibodies being made by an overactive immune system to a non-existent threat?
Sure.
Cross-reactivity is likelihood. It's likely, yeah.
I think the proof of the pudding will be in the sequences, and it shouldn't be that hard. So I think that's where we'll know if there's any reality to these things.
Sequencing is dirt cheap, and there are 100 companies that'll do it overnight.
Yep.
And as John Coffin has said, antibody results by themselves cannot prove a particular virus has caused an infection. You really need to get the virus.
Yeah.
Also, she writes, Mikovits continues, data presented in Lombardi et al. Suggested additional strains of gamma retroviruses and viral gag proteins could be directly immunoprecipitated from the blood of patients, supporting a finding that additional strains of gamma retroviruses were isolated. So Nicola writes, does the presence of these viral gag proteins constitute evidence of infection, or could they just be present due to contamination?
I don't know the data that we're talking about here.
She IP'd gag from patient blood. But so does that show that there's evidence of infection, I guess is her question.
I mean, probably not. I mean, if there's just a band on a gel of a gag, it says there's some protein there that's being detected.
That's the right size to be gagged. Yeah.
I think what they did in that figure, they co-cultured the blood with LN cap cells and then immunoprecipitated from that. So that could be a contamination there.
Sure.
Might be grown out from the blood.
Right. But I think it's still just in general, I think there's always still the potential there really are viruses out there, but we don't know that. And if there are, I think they'll be found.
And she wraps up by saying, personally, I think XMRV is dead as an infectious human virus. However, I think these are still interesting questions. I read Dr. Mikovits' letter carefully, and these were the two pieces of evidence that remain as questions in my mind. I am grateful for the work that you have done to explain XMRV. You guys are great fun, and I wish I had more energy to vote to TWiV. I have listened to quite a few non-XMRV podcasts and have learned a lot this way from all four of you and from your guests. Rich, if I ever get better, it is possible that you will find me knocking on your door asking to wash dishes. I'm not kidding. So Nicola is a CFS patient for the past 28 years.
I'm glad we can help out.
Yep.
All right, let's wrap it up with some picks of the week.
And there's plenty of dishes to wash.
You bet. Rich, what do you have for us today?
Let me find it here. Yes, I went to a lecture yesterday that was a physician's sort of look back on 30 years of treating AIDS.
Cool graph.
The physician in question was
This is HIV.
Judy Currier, who's the chief of the Division of Infectious Diseases at the School of Medicine in UCLA. And this graph is one of the graphs that she showed, which is trends in annual rates of death due to the 9 leading causes among persons 25 to 44 years old in the United States from 1987 to 2007. And the 9 causes are unintended injury, cancer, heart disease, suicide, homicide, HIV disease, chronic liver disease, stroke, and diabetes. And basically, with the exception of HIV disease, over the period from 1987 to 2007, the curves are all flat. But the HIV disease starts out at some number in 1987 and rises steadily at an alarming rate, cutting through all of these other curves to exceed them by about 1994, and then levels off, and then even more rapidly drops down to near a baseline by 1997 or 8, and then is still sort of petering out until the present day. And it is a really, really striking graph. And 2 things about it struck me. First of all, the way Judy presented this was her own look back on her clinical practice at this, and she did it in such a way that forced you to imagine what it must have been like to be a physician in 1993 or 94, seeing this thing skyrocket to take over all of the other leading causes of death and wondering where on earth this was going to go. And it must have been really scary. So that's one message. And the other is this amazing decline that happened over the years. And this is because of heart therapy. And the point I want to make is that science works. And it's government-funded science to a very large extent. And it's a lot of dedicated individuals who recognized that this was a serious problem and put a lot of energy and a lot of money behind it, and got the science together and have done really a miraculous job of bringing this thing under control. And I think it's just an amazing demonstration and a very heartening demonstration of what science can do.
Rich, I just want to second that and say this is such a great success story. When you look at it, it's really impressive. It's really phenomenal.
It's just the U.S. though.
All true.
Remember in Africa, the curve is still going up,
Yep.
right?
Right?
Yeah, no, nobody said it was cheap.
Yeah.
Nobody said it was easy, but it's still amazing that it can be done at all. And I think in the beginning, no one would have thought a virus disease could be controlled so successfully as this has been.
30 years ago this month, actually, the first CDC report.
Right.
Pneumocystis carinii in L.A. So that's pretty cool, Rich. Alan, what do you have?
Well, my pick of the week, I searched through the past agendas and realized we hadn't picked this yet, which surprised me. So in the off chance that there are TWiV listeners who haven't read it yet, this should be required reading for everybody, I think. It's Carl Sagan's book, The Demon-Haunted World. And if this doesn't end up being assigned reading for my daughter when she's in high school, I'm going to assign it to her. This is Sagan's book about how science works. And it's I think it's one of the best presentations of that's ever been created. The one thing that I would say against it is that it probably could have used a somewhat more rigorous editing. He does occasionally repeat himself and belabor a point, but the points that he's belaboring usually bear belaboring. And highly recommended. If you haven't read it, definitely.
Cool. I will.
If you haven't read it.
I'm gonna have to retire or take an extended vacation and read the whole TWiV list. It's an amazing collection of books. This looks great.
Have you read this, Steve?
No, I'm looking forward. I will try to do it. I love Sagan.
All right.
It's great. If I was picking journal articles, I'm going to give a
Sure.
journal club at my own group in a week or two. And I was fishing out for some of these fabulous papers that I don't know if you've covered or not, where people have found fossil DNA copies of old RNA viruses in the human genome.
Sure.
And Skalk has done some of that, and others. So viruses that you would never have thought would be available to you have been dug up in our own genomes. Viruses like Bornaviruses and others.
It's non-retrovirus. It's very cool, yeah.
Exactly. It's very cool.
So it allows us to date how old viruses are.
Yeah, millions of years. RNA viruses don't live very long, but they're hiding out as DNA in our genomes. So it's fabulous.
Cool. Great. Viral fossils. Well, my pick is a blog called Beyond the Human Eye. It is a blog written by Phil Gates, a botanist at Durham University in the UK. It's called An Insight into a Microscopic World Invisible to the Unaided Human Eye. So he gets water from here and there and looks at it. And you can see all sorts of interesting living things in the water. So that what's on the front page today are a few drops of water found in a pond. And you can see what are called desmids, these organisms. And if you go down, he's done this over and over again. Life in a few drops of water, diatoms, and all sorts of other things. Beautiful photos of them and nice descriptions.
Really cool.
Neat.
Yep.
We also have a listener pick of the week. This comes from Raihan. He writes, "Hello, strange voices I hear while riding to work. I would love to propose the following link as a listener pick of the week. Understanding the backlog of emails you guys have, I would anticipate that this would have been already picked, but I'm trying my luck anyway. The video link is a TED Talk about the eradication of poliovirus by Bruce Aylward, entitled How We'll Stop Polio for Good. The video is absolutely riveting. The pictures that Bruce shows in this talk are like scenes relived from another one of your previous picks of the week, smallpox, the death of a disease, the inside story of eradicating a worldwide killer. After watching this video, I can sense that the eradication of this disease is very near.
Really cool.
Neat.
Let's hope that the day comes real soon and Professor Racaniello will be out of a job and will be able to dedicate all his time for TWiV, TWIP, and TWIM. Keep up the amazing work, guys. Your humble parasite, Raihan from Singapore." All right, Raihan, thanks for that. And keep on listening. I don't know if polio's eradicated. That can work on some other viruses.
Yep.
So there you go, another TWiV, and this one is all about XMRV. And I won't say it's the last one we'll do on XMRV, but probably we won't do one for a while unless something happens, and we'll cover the Lipkin story whenever it comes out. You can find TWiV as usual on iTunes at the Zune Marketplace or at twiv.tv. Send us your questions and comments, as always, to twiv@twiv.tv. Steve Goff is at the Department of Microbiology and Biochemistry, two different departments here at Columbia University Medical Center. Thanks for joining us, Steve.
Happy to be here.
Great to have
Thanks,
you back.
Steve. This is really great to have you here.
Thank
for this
you.
one.
Appreciate it. Alan Dove is at alandove.com. Great to have you back.
Always a pleasure.
And Rich Condit usually is in Florida at UF Gainesville, but is hanging around Boston these days. Thank you, Rich.
On the road. Yeah, you're quite welcome.
And
Great fun.
I hear next week you're going to be in Texas, or is that the
That's
weekend?
correct.
Excellent. Looking forward to hearing about your adventures.
All right.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.